Hi, my name is Peter Kelmanson with Technical Information Services at the Jackson Laboratory. Thank you for viewing this presentation on Cre-lox Basics, an introduction to Cre-lox technology and mouse modeling.
Cre-lox technology has quickly become one of the most widely used and versatile tools for genetically engineering mice for human disease modeling. In this presentation, I will introduce you to the principles of the technology, outline basic Cre-lox mouse crosses, and demonstrate how to access the world's largest collection of Cre-lox mouse strains. Just a basic overview of what I will talk about. I'll first introduce the basic Cre-lox mechanism, and then I'll discuss the different strain types that are available and the various breeding schemes to generate tissue-specific knockouts, general knockouts, inducible knockouts, and then I'll talk about some Cre reporter strains as well. Then I'll finish up with a discussion on the various Cre-lox web resources that are available to help you find the right mouse strains, as well as information on Cre expression data as well. The Cre-lox system was discovered as a natural part of the P1 bacteriophage life cycle. The genome exists as a linear genome, and then following injection into the bacterial host, the phage genome then uses that Cre-lox system to recombine, form a circular DNA, which then is required for replication of the viral genome and packaging into new phage particles. So the Cre-lox technology was developed in the late 1980s, early 1990s by investigators at DuPont. Then it's been adapted for use in several different species, including yeast, plants, mammalian cell cultures, and of course what we're going to talk about today would be in mice. And so what's really great about the Cre-lox system is it allows for gene manipulation and then gives researchers the ability to alter and delete DNA, and then basically to regulate the location and timing of that gene recombination. So basically to create knockouts or other mutations when and where is important for your research questions. The Cre-lox system is a relatively simple two-component system. The first is the Cre recombinase, which is a site-specific enzyme that's going to catalyze DNA recombination between two different LOXP sites. And for the presentation, we'll just signify it by this little scissors diagram here. And then the LOXP site is a 34 base pair DNA sequence where it has sort of this 8 base pair core sequence flanked by 2 inverted repeats. And the location and orientation of those LOXP sites will determine the recombination result. The most common use of Cre-lox system is for deletions, but inversions and translocations are available as well. And the Cre-lox system allows for a very abundant possibilities for genome manipulation. So, as I said, the most common use for Cre-lox is for deletion, and this is a diagram that sort of outlines how it works. The two components here are a LOXP flank gene, or a portion of a gene of interest, where those two LOXP sites flank that gene of interest, and they're in the same direction. That sort of a strain is created the same way any targeted mutation would be created. Then the Cre recombinase is essentially going to recognize those two LOXP sites and then is going to essentially recombine the DNA between them, leaving the DNA in between the two LOXP sites out as a circular piece of DNA and a single LOXP site remaining on the chromosome, thereby knocking out the gene. And then it's the promoter, or however the Cre itself gets expressed, that controls when and where this deletion happens. If the two LOXP sites are put into the opposite orientation, so they're facing opposite, then what the Cre recombinase does is that will actually invert the DNA, so it essentially flips it over. Not as common a use, but there are a lot of really relevant uses for doing such a thing. If you have the LOXP sites on two different chromosomes, then you can use the Cre-lox system to generate translocations, where the Cre then recognizes the two LOXP sites and then recombines the DNA, thereby translocating the two arms of the chromosome. It can be used to sort of mimic those sort of chromosomal aberrations that you find in human diseases, like some cancers and some developmental issues as well. So probably the most common use for Cre-lox system is to generate like a tissue-specific knockout, used to really study the role of a specific gene or protein in a specific tissue or cell type, or to get around the issues of, say, embryonic lethality, where a lot of genes or products that you produce, if you knock it out in the entire mouse, can be embryonic lethal. By regulating or knocking out only in certain tissues or at certain times, you can use Cre-lox system to get around that issue. And some of the nomenclature that you'll hear is where you have a gene that's flanked by 2 LOXP sites. A lot of people call them floxed or flanked by LOXP. And just in case, I want to make you aware of that, just sometimes I use either flanked by LOXP or floxed when I go through this. So the first cross is with generating a tissue-specific knockout is you cross your homozygous flox mice to your Cre strain of interest. In this case, it's an albumin Cre, which gives liver-specific expression of Cre recombinase. And from that cross, you'll generate mice that are heterozygous for the floxed allele, so they have one floxed allele, one wild-type allele, and then carry the albumin Cre. So in these mice, the whole mouse itself is normal, but in the liver, the Cre is expressed, and therefore then the LOXP flanked allele can be deleted. However, you may notice that we still got one wild-type copy, so we haven't actually completely knocked that gene out in the liver. We've only knocked out one copy. So therefore, 2 crosses are needed actually to generate the mice you want, and a lot of researchers are not aware that it would take 2 crosses. So in this case, we'll then take this mouse that's heterozygous for the LOXP flanked allele, carrying the Cre, and what we will do is then we'll take that mouse and cross it back to your homozygous flox mouse strain. And from that, you'll generate mice then that are homozygous for the flox allele, carry the albumin Cre. If you want to know, it'll be about 25% or a quarter of the mice. And therefore, in those mice, then they have 2 copies of the LOXP flanked allele in the liver. Therefore, the Cre can knock out both of those copies, thereby creating a knockout in the liver only. So at the Jackson Laboratory, we do get a lot of questions on how to set up the breeding and calculating the percentage of the tissue-specific knockout mice generated. So I just want to go through how that works. And really the easiest way to do that is to use the old tried and true method of a Punnett square like this one to calculate the percentage of the tissue-specific knockout generated. So from this mouse right up here, you're going to get 4 different possibilities. So you're going to get mice that will have the Cre, or won't have the Cre, and then you'll get mice that will have the LOXP flanked allele, or they'll have the wild type allele. And then when you put those 4 possibilities, and then you cross them to the homozygous LOXP flanked mouse, which can only provide the LOXP flanked allele, you'll get 4 different genotypes out in approximately even ratio, 1 to 1 to 1 to 1, and therefore then the 1 out of the 4 that you want, or the 25%, that would give you then the homozygous floxed with that albumin Cre. Now, realize that there are like free Punnett square generators online that you can use to use this sort of calculation for you, so you don't always have to draw it out yourself. All you have to do is really Google Punnett square calculator, and you should find them. So if that helps you with these crosses, I recommend you take advantage of those tools to help you with your crosses. So I'm just going to take a quick step back and show you an alternative way to get a liver-specific knockout. So I've just talked about crossing these mice that are heterozygous for your LOXP flanked allele and het for the Cre, and breeding them to the homozygous floxed mouse. However, you may find in some cases in the mice that are homozygous floxed with the Cre that the Cre may not as efficiently delete both copies of the floxed allele as you would like. And so one way to improve that efficiency is to then actually breed this first generation mouse to, say, a heterozygous null, like a complete knockout in all tissues, is to substitute that instead. And from that, then you can generate mice where then you have one LOXP flanked allele and one complete knockout allele, as well as having the Cre. And then what that will do is then the Cre recombinase only has to knock out the one floxed allele in the liver, thereby giving you a liver-specific knockout. And from this cross, you would expect about 12.5% of the mice to have the genotype of interest. So if we go back again to our Punnett square and fill in the traits, again, the 4 options for this mouse up here by the heterozygote, one wild type and one knockout allele. There you'll see the 1 out of 8, or the 12.5%, that are the genotype that you're looking for. However, this cross will also produce mice that are heterozygous for the Cre and heterozygous for the knockout allele. You can actually take those mice to help get the mice you want for future breedings as you're expanding your colony. So if you can take those mice, which now is here, your albumin Cre and het for the null, then cross that to your homozygous floxed mouse, you'll actually then generate 25% of the mice that are het for the floxed allele, one floxed allele copy, one knockout allele copy with the albumin Cre. I'm not going to go through the Punnett square, but if you want to confirm that, go ahead and draw that out just to confirm whether I'm correct or not. Okay, so let's move on to how the Cre-lox system can be used to generate germline or whole body knockouts. And this is accomplished by breeding your floxed mice, again here, floxed mice of interest. In this case, a germline specific Cre. In this case, it's a ZP3 Cre, which is expressed in the zona pellucida, so it has oocyte specific expression. So therefore, it's only going to be expressed in the female germline. Cross those mice to generate mice that are now het for the floxed allele and the wild type allele and carry the Cre. Then what's going to happen then in the ovary as the oocytes develop, the Cre is expressed and then can recombine and create the knockout. And then therefore those females will then pass along a mix of wild type and the recombined allele. You will still need a couple more generations from this point to actually produce a homozygous null mouse. And so one way to speed that up a little bit, a more efficient way is generating a germline knockout is to use a Cre that's sort of driven by a very early and widespread promoter. For example, like the SOX2 Cre. It essentially does the same as a germline specific, but it can get away with having one less generation of breeding to get your desired animal. So in this case, again, you breed your Cre strain, SOX2 Cre, to your homozygous flox mice of interest. And that will then generate mice that are heterozygous now for the recombined allele, as well as carrying the Cre. And then you essentially cross those together to generate 25% that would be homozygous for the knockout. Sometimes we do recommend that you may want to breed these mice to a wild type first, just so that you can verify recombination did occur in the germline. Also lets you get rid of the Cre, because once it's in the germline, you don't need the Cre anymore, and you can breed that Cre out to get rid of some of the issues or off-target effects of having Cre expressed in high levels in all tissues. I'll mention that again in a little bit as well. Just a summary here to compare the differences between a Cre using Cre-lox to generate tissue-specific knockouts versus germline knockouts. Both of them require at least 2 generations of breeding. Depending on the germline or embryonic deletion strain you use, it could be 2 to 3 generations of breeding. For the tissue-specific, the Cre is required to maintain the line for future generations. You always have to have, in this case, the albumin Cre present every time to be able to knock out that gene in the liver. If you get rid of the Cre, you won't have the knockout in the liver. For germline, once the Cre does it in the germline, then the Cre is not required after that deletion, so you can get rid of it. And so in the tissue-specific, the genotype of your whole mouse is going to be homozygous flox Cre positive. But in the tissues where the Cre is expressed, you'll be homozygous flox deleted Cre positive. The genotype of the whole mouse for the embryonic or the germline deletion is all of the cells and organs and tissues are homozygous for the flox deleted or Cre recombined or knockout of what your gene of interest. Now, I've talked about tissue-specific and germline Cre, and I just wanted to let you know that there are a lot of recent advances in Cre-lox that actually allow for temporal regulation of Cre recombinase using an inducible Cre. In this case, which is the most common sort, the Cre itself is fused to a modified estrogen receptor, such as ESR1, which will bind to tamoxifen, but not to the endogenous mouse estrogen itself. And what that does is then it prevents the Cre recombinase from entering the nucleus, which is required for it to then recombine the DNA to get rid of your recombine the floxed allele. It keeps then the Cre in the cytoplasm until the tamoxifen is given, which point it then can enter the nucleus and recombine the DNA. So therefore, these can be used to generate knockouts in either the whole mouse or in tissues at very specific times. So you can look at what a knockout happens when you knock out a gene in the first week versus in a 6-month-old mouse or whatever is working for your experiments. So the breeding for the inducible Cre is exactly the same as a tissue-specific Cre. You cross the 2 strains together and then in 2 generations to generate homozygous flox mice with the tamoxifen-inducible Cre. In those mice, they're going to be normal, but then when the mice are then given tamoxifen, then the Cre can then recombine and knock out that gene of interest. So I would like to talk about an analogous system to Cre-lox. It's known as FLIP and FRIT. This was derived from a yeast. It is becoming a little bit more popular in mouse-based research. And essentially, there's a FLIP recombinase that recognizes a pair of target sequences, or FRT, that find your genomic region of interest. It doesn't work quite as efficiently as Cre-lox, and in fact, it's often used in combination with Cre-lox because Cre does not recognize FRT sites and the flip does not recognize the LOXP site, so they can be used together. The most frequently used is in the generation of the original LOXP flanked allele. So here's your LOXP flanked allele where they're flanking, in this case, an exon in your gene of interest. But to do traditional targeted mutagenesis to generate those strains, you have to have a selectable marker, typically a neocassette that gives neomycin resistance to that cell line. But then what one can do is then flank that neocassette with FRT sites. Then once those mice are generated, they're crossed to, say, a germline flip to then remove the neocassette and have just an single FRT site remaining. That way this neocassette doesn't interfere with the normal expression of your gene of interest. And now you're left with just this LOXP site flanking your exon, a single FRT, and then you cross that to your Cre strain of interest. It's a very common usage for using the flip-frit system. As with all things, there are going to be some considerations for using a system like Cre-lox. Realize there can be some sort of mosaicism. Some of the target cells that you're looking at may not express Cre, or the LOXP sites may not recombine. Every floxed allele can recombine with the same Cre with a different efficiency. So here you can see in this picture some mosaicism in mouse mammary gland epithelial cells where after crossed to a Cre strain. There can be integration site specific for your Cre expression, so even though you have the same construct, different founder lines can have different expressions. So if you're generating your own transgenic Cre mice, you definitely need to evaluate multiple founders. You can also get expression in unexpected tissues, including the germline. So you may have a tissue-specific Cre, like say a heart-specific or a liver-specific, and it could be also expressed in some off-target sites that you're not expecting, and germline or other tissues that you don't expect, and I'll show you some examples of that as well. So therefore you may want to use either Cre-positive males for breeding, because that tends to happen less in the males, but you can also use a reporter for test expression, and I'll talk about those in a moment as well. And then, of course, the efficiency of LOXP site recombination can be affected either by the position where on the chromosome those LOXP sites are. Certain portions of the genome have easier access for Cre2 LOXP sites than others, as well as the distance between them. And then, of course, for breeding, it's best if you have your Cre transgene on a different chromosome for your LOXP flanked allele. If they're on the same chromosome, you would have to get a recombination event to put them on the same chromosome to be able to get homozygous flox Cre positive mice. And again, if you are using germline Cre for germline deletion, it's a good idea to breed that Cre transgene out at some point so that it doesn't interfere with the phenotype that you're looking for. As Cre can produce a phenotype by itself, it can either be due to where the Cre has inserted in the genome, giving it either a phenotype. It can alter gene expression in the gene it may have entered in or nearby, and there also can be some Cre toxicity where it just has some sort of toxic effects as well. Also realize that you may want to pay attention that look very carefully at your target gene and when it's expressed, especially during development, as compared to when the Cre is expressed, because if your target gene is turned on and does what it needs to do before the Cre is ever turned on, well, you may get recombination, but the gene has already done what it's supposed to do, and you may not get phenotypes that you're looking for. And as with all things, do keep in mind that genetic background can affect the phenotype. Okay, so now I'd like to move on, switch gears a little bit, and talk about Cre reporter strains. The reason that people have generated Cre reporter strains is a great way to be able to visualize where and what tissues and organs Cre is expressed. That way you can either confirm that it's expressed in the tissues you want and not expressed in the tissues where you don't want it expressed. So the Cre reporter strains are typically generating using what's called a lock-stop-lox, or sometimes an LSL. And what that is that you have a promoter, usually a strong, widespread promoter, like a chicken beta-actin or a GT-ROSA 26SOR, driving some sort of molecule like LAC-Z or a fluorescent protein. But then following the promoter is this LOXP flank stop cassette, lock-stop-lox, LSL, right, which prevents expression of LAC-Z. In the presence of Cre recombinase, that stop cassette is removed, and then LAC-Z expression or whatever your molecule then gets expressed, and you can visualize that. And then you can use that to assess Cre activity in your tissue or tissues of interest. And conveniently, for using Cre reporters, only one generation of breeding then is required. And here's just an example of showing LAC-Z expression following widespread Cre recombination. So here you can see a control mouse. There's no LAC-Z, but the entire embryo here is pretty much blue. And here you can see some sections then showing that widespread blue expression. So as I mentioned, only one generation of breeding is required. So here you have a nest-in Cre, which gives you some neural specific, but this could be any Cre strain, cross to your Cre reporter, your flock stop, lox, in this case LAC-Z. One generation of breeding to generate mice that are carrying both the Cre and the Cre reporter. And then in those mice, then you look for, in this case, LAC-Z staining. Here you see it around here, to confirm Cre expression or Cre activity in the expected tissues that you want. However, this same sort of principle can be used not just to use a Cre reporter, but to actually create certain disease models. And here's one example where instead of using, say, a LAC-Z, there's this KRAS G12 oncogene, which now has expression blocked by the LOXP flank stop cassette. But then when Cre removes that, then that KRAS G12D oncogene is expressed in those tissues, leading to tumor formation. So, for example, if one used like an MMTV Cre, so that then has expression in mammary tissue, cross to this KRAS G12D flock stop mouse, then in mammary tissue you'll get that deleted expression of that oncogene, and then those mice will develop mammary neoplasms. And only one generation of breeding is needed for that. So here at the Jackson Laboratory, we do have the largest selection of Cre-lox mouse strains available in the world. Going back to April 2014, we have over 425 strains that express Cre. Of those, 170 plus are inducible. We have over 730 strains that carry some sort of flox gene, and more than 175 strains that have a LOXP flank stop cassette that can be used as a Cre reporter. And then we all have more than 200 that have neuronal specific Cre expression, and you can go to that link right underneath there to get a list of those strains if you're interested in neuronal expression of Cre recombinase. So we do have a Cre repository page, which you can find at the link you see at the top of the page. This is dedicated to our Cre-lox repository, which can help you find the Cre mouse strain that you're looking for easier. And what you'll see is we have this lists of available Cre strains, and if you just follow that link, we'll pull up this page, which then has some categorized Cre and flox strains, as well as all of our flip and FRT flanked ones as well. You can see at the bottom, and you can click any of those links to pull up an appropriate list of strains to look through, and the list will look something like this. And what you can do then is you can say, just in your internet browser, do a text search for a specific term, like say liver, and then do a search, and then it will pull up all of those keywords on this page, and then you can find the liver specific Cre, or kidney specific, or heart specific, or whatever you're looking for. And in fact, that you could have used to find the same albumin Cre strain that we had talked about earlier and used in some of the examples. And this is a live page, and it is curated and up to date when we import new Cre strains as well. And for some of our more popular Cre strains, we do have some Cre expression data that is available online, and you can find it right off the Cre repository main page, showing the link with the arrow right there. And this will then pull up a list of all of the strains that we have data for, alphabetized by the promoter. So you can see the albumin Cre is first. And this is sort of what it looks like here. And then what you do is if you click on the picture, then you'll see all of the data that we have, and we look at 4 different developmental time points. And anything that's relevant, then you'll get a picture, and of course you can click on the pictures to get a better view of the data. Here again, using the LAC-Z reporter, you can see Cre expression in this strain in the lung, the heart, the skin. As you can see here, we collect Cre expression data from 4 different time points: embryonic day 10.5, embryonic day 15.5, as well as P7 and then P56 mice, which would be like an 8-week-old adult mouse. We do full necropsies, and we collect various 30 different organs and structures, and then you can use that nano zoomer slide scanner to look at that data up to 20-fold. And then this data is also shared with the MGI Cre portal database, which is a database that is maintained also at the Jackson Laboratory using that Mouse Genome Informatics, or MGI database, which is one of our other Freeline online resources that you can use to find Cre-lox strains. But in this case, this Cre portal not only has data from Cre expression for strains maintained by the Jackson Laboratory, but also others that are out there reported in the literature. So if you can't find what you want at the Jackson Laboratory, you can use this Cre portal to search for other sources of strains, and you can either search by the promoter or by the site of expression. For example, you could type in muscle and look for strains that have Cre expression in muscle. And if you do that search, then you'll get a list of strains like you can see here. All of them have muscle, expression in muscle, as well as, again, these also have muscle where no recombination is detected. So you can use it to select that way. And here you can see in this one, it's an actin 1, alpha 1 Cre here. And then if you click on that, then you'll get more information, and you'll see that the expression level is in muscle when the mice are an adult. But you could also see if in other tissues that it's also expressing as well. And here you can see then sometimes you can even get some pictures of sections showing Cre expression, especially where those strains are held at the Jackson Laboratory, and then we have collected that expression data. Then it is uploaded right there on this Find Mice IMSR line, and you can see that there are 2 mouse strains that are commercially available. And if you do click that link, you will then pull up this page, which shows you more information on those strains and where they are available from. And again, this is not just mice that are available from the Jackson Laboratory, albeit these strains in question are, but you'll also find mice from other repositories in the United States, Canada, Europe, and Japan, and Australia. Going back to the Cre portal, you can also, when you did the initial search, you'll actually see there's that Find Mice IMSR column where you can actually then just directly click right on that number 2 and then pull up those strains. So you can use that to help you find strains that are commercially available and use them. This presentation was made possible with your support. Jack's Mice and Services is dedicated to providing you with quality resources and flexible solutions and innovative technologies and unsurpassed expertise to support all aspects of your research. And realize that every dollar that you do spend at Jack's Mice and Services does support our shared research goals and sustains all these valuable mouse models and resources that are only available from the Jackson Laboratory. We do offer a host of services that might be helpful to you. Besides just having the specialty strains, we also do basic, custom, and complex breeding capabilities. So if you're having trouble breeding or you wish, we can breed Cre-lox mice together for you and either send you the breeding pairs or maintain cohorts for you here, as well as doing compound efficacy testing and genome scanning as well. And with that, again, thank you for attending this presentation. And if you do have any other questions, you can contact technical support at this link here, or you can contact your regional rep as well today if you want information from them or schedule a visit if you'd like to get them in and talking to you directly.